Hello, this is Gene Gallagher. I'm going to make a brief movie that should take no more than about 5 minutes, showing you the major result of a canonical principal component analysis of hypergeometric probabilities, or C-PCA-H, done by Angeliki Evgenidou, and that will be presented in her November 13th, 2012 Ph.D. Okay, we're since 1992, the Massachusetts Water Resources Authority has been sampling the infaunal benthos in stations near the MWRA outfall. This outfall was constructed and it went online September 2000. So these are all the near field stations. They're sampled every August. Many of the stations are sampled with 3 replicates, and then some of the stations are just sampled with a single replicates. These are the far field stations, and here is that MWRA outfall. The basic idea behind the design is that it is a backy design, before-after control impact experiment, where the effects of the outfall will be assessed by examining the changes in community structure in the near field and the far field before the outfall went out, and then assessing whether the differences changed after the outfall went in place in September 2000. In this analysis, we will have 3 years of post-outfall data, 2001, 2002, 2003. I'm just going to, we'll see all of the stations, but I'm just going to focus on 4 stations from each data set. NF17 is located very close to the outfall. It's a sandy station, very little silt clay, and it's dominated by 2 building amphipods. Near field 2 is a station that is next to a sandbank. So it's an unusual station in that it was originally silt clay when first sampled, and then strange things happened to it. Here is NF8, which is a station also near a sandbank, and then the other station that I would like to focus on is NF12, which is a near field station in here. And then, well, here's far field 12, that may pop up in the movie. The far field stations is always a major concern, particularly to the residents of Cape Cod, whether the MWRA outfall would affect Cape Cod Bay. So we'll look at the effects on 2 Cape Cod stations, far field 6 and far field 7. It's very important stations. The deepest station in the data set, about 60 meters deep. This is far field 4 in Stillwagon Basin. This is the Stillwagon Bank, which is about 30 meters deep. This region is the most, by many accounts, it's one of the most important whale watching areas in the world. There are more species of whales that come to feed in this area than just about any other location on Earth. And then we'll also take a look at Farfield 9, which is a station that is located to the southwest of the outfall, and this is a direction of one of the dominant current patterns, which basically move around this region. So let's take a look at what these stations looked like after August 1992. And this is a C-PCA-H ordination diagram. This diagram partitions the variation in CNES. CNES is an index developed by Gallagher in 1992, published with Trueblood 1994 in Limnology and Oceanography. It stands for Chord Normalized Expected Species Shared. 2 samples. You evaluate the species composition of 2 samples by drawing a fixed number of individuals from each sample. And in this case, 15 individuals were drawn from 2 samples. So you would draw, say, 15 individuals from NF17, 15 individuals from NF15, and calculate the expected species shared. And then you would normalize this expected species shared by the diversity within each sample. How many species would you expect if you drew 15 individuals from sample NF17, put them back, and then drew another 15 individuals? And then using a metric, an equation that produces a metric index, you can plot the CNES distances in space. You can get a graphical display of the CNES distances. So the basic technique is called PCA-H. Angeliki Evgenidou has developed a very important modification of it called canonical PCA-H, where you put a constraint on these ordination diagrams that they must be linear functions of measured environmental variables. And these include grain size, total organic carbon, depth, and in this case, The indicator variable, year is another variable. In this case, the indicator variables indicating near field and far field were also included, as well as an indicator variable indicating pre versus post, so pre-outfall samples and post-outfall samples. So you can see here, okay, this is the major, this is C-PCA-H axis 1, C-PCA-H axis 2. The major source of variation is distributed along C-PCA-H axis 1, which Angeliki has found to be strongly associated with depth. The second axis is, the most important determinant of it is grain size. So these tend to be high silt clay samples with a negative PCH axis score. And then the sandier samples, and NF17 is a very sandy sample with very little silt clay. And that plots up in this region here. So this is the pattern in 1992. Here are the NF2 station, NF8 station. They're very, very similar in species composition. The size of the circle in this diagram indicates the position of these samples in a third dimension, so that the larger symbols are located in the, pointing out of the display, and then the smaller symbols are pointing into the display. So there is pattern in 3 dimensions. The actual dimensionality of this display is 360 dimensions, because there are 361 samples being plotted in this display, and there are roughly 370 species in this data set. So let's go through. This is using the Google Trend Analysis, which is based on Hans Rosling's Gapminder software, and we will go through and let's focus on a couple of stations. First time through, we'll focus on NF2, which is a station that started out at about 80% silt clay, and then the station location remained the same. It was located by GPS, but a sandbank covered that original sampling location in the original community. This ordination diagram is based entirely on changes in species composition, not on grain size, although grain size is used to basically reorient the display so that the changes in community structure that are most strongly correlated with environmental variables are displayed. So let's start the display, and then when we get to 2000, 2001, that is the key period that we want to do. I'll make sure that I turn off trails. I can actually track these trajectories, but they get a little noisy. So let's start. Okay, what happened to NF2? There goes NF2. It's zooming up. So it's zooming up. It almost got up to NF17 as it went from dominant silt clay to sand. Here's NF23. Here's a station that got very sandy, and then it's not so sandy here. Here are the 2 Cape Cod Bay stations. They're relatively stable, 1996, 1997. These stations are relatively stable. Farfield 9, relatively stable. We're heading up to the 2000 period. So it's 1999, 1999, 1999, 1999, 2000, 2001. Did you see the transition? The samples split apart. So that the 2000 to 2001 transition, when the outfall went up, was like a speed bump, where these samples moved a little bit to the left, these samples moved a little bit to the right. Let's go back and take a look at that transition. We'll take it back to about 1999, and then we'll start up the display again. So 1999, 1999, 2000, boom. All of these samples, the differences between the near field and far field got more extreme during that period, between August 2000 and August 2001. Now, this was a statistically significant change in the data set using the permutation methods from Kanoko. It's an ecologically significant change, but in terms of environmental management, it was not a harmful change. Community structure changed. We could detect the change. We could detect it. We know, Angeliki Evgenidou has documented exactly which species are responsible for the change. There are 3 species, a capitellid polychaete, Mediumastus californiensis, that increased in frequency in the near field areas. There's 2 other species that increased in frequency in the near field after the outfall went out, and those were Ericidia catharina and Theryx acutus. Ericidia catharina is a perionid polychaete that is a subsurface deposit feeder, perhaps a diatom feeder, and Theryx acutus is an interface feeder. It's common in Boston Harbor, and it increased in frequencies and samples out in Massachusetts Bay. So let's run through this once more. We'll see the changes in community structure due to grain size. So here is NF2. This is the sandy site going up to the silt clay site going up to a sandy site. Here's the NF17, the sandy crustacean two-building community. It's fairly stable during this period. Here are the Cape Cod Bay stations. Farfield 9, which is a station that's fairly near to the outfall. We're heading up to 1999, 2000. There we see it, the transition from 2000 to 2001. It's almost like there is a magnetic repulsion between the near field and far field stations as they diverge slightly after the outfall went out.